For the previous editions of the Pharma Day, welcome all of you, scientists, clinicians, and patients in a few days through YouTube video to discuss about myotonic dystrophy. So first, an update from Euro-Dyma. This slide is the last slide that I presented during the Euro-Dyma CRN meeting in Munich last year, not this one, the year before. And the conclusion, where I have presented all the aspects of myotonic dystrophy in the patient point of view in Europe. And the conclusions from this talk was that there was a considerable lack of understanding, a lack of cognitive and GI disaster knowledge, and also a lack of care for outside of a very, very specific clinical point, which are neuromuscular centers. So what happened from last year? Some events in Europe. I have asked all the members of Euro-Dyma to provide one or two slides, one or two events having take place last year or this year in Europe. I had many of them, so he was obliged to make a selection. I don't want to keep the floor for long. So the first one was the Sympathetic program. Sympathetic is dedicated to repurposing. It is still ongoing up to 2028, and it's working. Patient engagement is very strong. We are connected to train people, train patients and family to adopt repurposed drugs. Last year also we had the ENMC meeting dedicated to gastrointestinal aspects. This was a really important meeting where the clinicians, we had many gastrointestinal experts also, to discuss symptoms, treatments, and the future work to be done in the domain of gastrointestinal, which is really an unmet need for myotonic dystrophy. I encourage you to look at the poster number 13, which is fully dedicated to this program. Also, from the patient side, we participated in, we helped the diagnostic group from DGM to be reshaped, and they had the first family meeting in Germany after a long time of nothing, with more than 120 people on board. Of course, the Euro-Dyma CRN, which is still under construction. DM1 Hub in Spain, really impressive program starting with a new registry. AFM made a family meeting dedicated to myotonic dystrophy. We had more than 200 people connected through video. For Entry-DM also, which is a nice program shaped by Mario Gomez in France and in Europe, it's dedicated to demonstrate to young scientists having just finished their school time, and we will participate in DM studies in the future, how to deal with myotonic dystrophy. And from Cure DM, a family meeting in 2 days with more than 170 people on board. So if you look at this, it means that Europe is moving, and it's not the worst news for this coming time. So for the mission and partners of Euro-Dyma, well, it's, you know many of this. The missions are to improve awareness, to bring the voice of patients, to address unmet needs, as I mentioned, and connect with all the actors in the DM field. And for the list of participants, of course, we have the European patient organizations. We have also a very strong connection with MDF. We have a connection with drug industry through a program that we have starting two years ago, which is called the Pharma Dialogue Group. It's a group of a subgroup of Euro-Dyma made of people approved by their patient association, labeled by them, in charge of discussing with pharma in a very strict non-disclosure agreement program. So if we have fixed the fee to participate at a very low level to make sure that money is not an issue. So if pharma need to share with people, with patients approved and validated by patient organization, it's a good way to do. And we have now a connection established with EMA, and it's very important that our association is also connected to the very last point where the patient voice can be heard, which is before the delivery of the drug to the countries. And unfortunately, we have a limited amount of resource to do that. So last year, here is the composition of the team of Euro-Dyma. And it's a good news that for this year, we have added 3 more. Ireland is joining us. Muscular dystrophy Ireland is joining us. We also have Portugal with APN, the Neuromuscular Association APN, who joined last year. And the second association in Spain, which is ADMES. These 2 associations are small associations, but very well focused and engaged in myotonic dystrophy. And that's it for me. Have a nice meeting.
So welcome from the Myotonic Dystrophy Foundation as well. Thank you, Alain. We have really a wonderful working relationship. So I want to make this really quick because you are here not to hear from us, right, about us, but you want to hear about progress in pharmaceutical development. So a little bit about the Myotonic Dystrophy Foundation. Our vision is, of course, that we want to see a world with treatments and cure for myotonic dystrophy. The mission of the foundation is community, care, and cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care. And we accelerate research towards treatments and cures. And that's why we're here today, right? That's the whole idea. So the organization was founded in 2007. It's the largest DM-focused patient advocacy organization in the world, and we serve tens of thousands of patients and families in the US as well as internationally. Let's pivot. We want to break down barriers towards treatments and cures, and that's really the ultimate and best indicator of progress and hope. That's and I encourage you to visit our new website with the newly organized overview of clinical development programs. So we have over 20 known or publicly disclosed programs that focus on myotonic dystrophy, and around 11 of them are in some form of clinical testing phases. To end here, that's our program. So today we engaged with all of you, with the majority of industry partners, and we asked, do you want to participate? And if so, in what forum? So we have long and short presentations. You see the presenters here. In the first block, we hear the short presentations, and then later the longer presentations. And then we are closing, we have a coffee break, and we are closing with a lunch. And for lunch, please, we are overwhelmed by this attendance here. Please only attend if you RSVP'd.
Good morning, everyone. Let me see. Okay, perfect. So my name is Emily Fantelli. I'm here from AMO Pharma. We're a small biotech company based out of the UK. We have less than 10 employees, and we have been in the DM1 space for the last 10 years. I've been attending IDMC since 2017, so it's nice to see a lot of familiar faces here. Thank you to Euro-Dyma and MDF for giving me the opportunity to present this morning. I don't need to tell anybody in this room what DM1 is. This slide is really just to highlight that our focus has been on the congenital form, and that's where you see our research has been focused for the last 10 years. Our drug AMO-02, or titiglucib, has a dual mechanism of action. It is a GSK3 beta inhibitor, which works a little bit upstream of where the CTG and CUG repeat problem is in individuals with DM1. And what we've found is that we also work directly on the CUG repeats to decrease the number of those. As I mentioned, we have been in the clinical trial space in DM1 since 2018. We ran our first study out of the University of Newcastle in 2018, and it was an open label proof of concept study. We did 2 cohorts where each cohort received a different dose level of tidagliflozin. And with the efficacy findings from that study, we went ahead and moved into our phase 2-3 double-blind trial for children with congenital age 6 to 16. We ran that study globally, and in that study, we completed 50 individuals. We recruited 53, completed 50 individuals with about 5 months of treatment, double-blind study. And after that, everyone who completed was given the opportunity to roll over into an open-label extension phase. So the double-blind portion ran from 2021 to 2023. And so our first individuals that started rolling over into the double-blind portion began that in 2021, and many of those continue to receive treatment in the open label portion today. So we had a total of 65 patients enrolled in the open label portion. You might say, Emily, you only had 50 people in the initial study. How'd you get 65? There were 2 ways into this. You could either roll over from the original double-blind study, or you could enter as a patient who hadn't received treatment before as a treatment-naive participant. And the study design for this open label was initially to offer 52 weeks, but we've continued to have extensions to that. And as I mentioned, some of these individuals have been on open label treatment since the fall of 2021. What you can see here is how long we've had individuals on treatment. Any of the yellow ones completed at least a year of treatment, if not more. Over the last 4 years, we had one site that got very busy with their clinical rotation and weren't able to continue. And then we obviously have some folks who have chosen to withdraw from the study. But for the most part, our retention has been fantastic. And I think what's really encouraging to the community here is you see a patient community that is willing and ready to be in clinical trials and ready to be a part of the research that's out there to offer. From our original double-blind study, we completed 50 participants. 49 of those rolled over into the open label extension. They opted to continue the open label treatment. And of those 46 of those individuals completed a full year of treatment, and 40 of those are ongoing. And then from our treatment-naive patients, we enrolled a total of 16. 13 of those completed 52 weeks of treatment, and then 11 of those decided to continue the access beyond the 52 weeks. And again, this is just really to highlight the fantastic retention that I feel we've had in these studies. In terms of demography of who's participated in these studies, we've had 49 participants from the original double-blind study. That was only for patients with congenital myotonic dystrophy age 6 to 16. So when we opened it up to treatment naive, that gave the opportunity for individuals who maybe didn't initially qualify for that study or didn't have the medical records to verify congenital, as we opened it up to both congenital and childhood participants. So we have the treatment naive participants, 7 of those qualified as congenital, and 9 of those are considered childhood onset. We also increased the age range in our open label treatment study. So this does include some young adults and adults with both childhood and congenital form. Adverse events is what you would expect to see over following anyone for 4 years. Most of our adverse events have been mild in nature, most of them unrelated. And in terms of what it looks like, anything that you would see in a 6 to 27-year-old. We do know that our drug, in about a third to just under a half of participants, can sometimes experience a rise in ALT and AST. This comes back down on its own. Oftentimes medication adjustment isn't needed. This is something that spikes, resolves, comes back down. So we have protocol in place to handle that if that happens to participants. Doesn't happen to everybody. Other than that, you just see sort of run-in-the-mill AEs that would occur in any clinical trial. And in terms of how we're measuring efficacy, one of the rating scales that we have in this study is called a clinical global assessment of improvement. It's a 7-item Likert scale, where 1 through 3 are gradients of improvement, 4 is no change, and then 5 through 7 are gradient of decline. And anywhere that there is purple on here indicates at least a minimally worse. And so what you can see is that as we get into weeks 164, 180, 196, 212, we don't have any instances of anyone worsening, which, considering that's a pretty long time in a clinical trial for some pretty sick kiddos, we're happy to see that. In general, the day-to-day tolerability of AMO-02 has been very tolerated, excellent profile there. We do have a DSMC that looks at the data quarterly. Additionally, over the years, we've always corresponded with regulators about our findings. The instances of increased LFTs, something we anticipate and are monitored and managed with dose adjustments and protocol management. And then we also have high retention in what we see as favorable CGI-I scores. In terms of our next steps, we've spent the last 18 months in lots and lots of discussions with regulators about the data we've collected thus far and what we can do moving forward. We're nearing completion on getting kind of final readouts from the regulators on what our next plans would be. We do remain committed to the development of AMO-02 in DM1 and specifically in congenital. And we're hoping to be able to announce later this year our next steps as agreed upon by the regulatory bodies. And that's everything. Thank you.
Okay, good morning, and thank you for the opportunity to present today. So we are very excited to talk to you a little bit about SRP-1003. And we've had the pleasure to start working with many of you, and for those that we haven't, we wanted to just quickly introduce Sarepta. So Sarepta is a global biotechnology company, and we utilize precision genetic medicines to treat rare diseases. And what we have is we have 3 treatment modalities. So this includes the siRNA that we'll be talking about today. We also have 3 RNA-based therapies, and we have a gene therapy. We have 4 products that have been approved on the market, and these are all in Duchenne muscular dystrophy. These products were some of the first disease-modifying products that got approved in Duchenne muscular dystrophy. They started with our RNA-based therapies, and they were really the first to offer some disease-modifying potentials. We also had the first gene therapy that was approved in Duchenne, and this really helped to alter the course of disease. Each of these products has been administered to thousands of patients, and behind each one of these, we of course know that every patient has their own unique journey, their own unique challenges that they went through, and Sarepta has always employed a patient-first approach. So we're very excited to take that patient-first approach into DM1. We're really thankful to learn from the community, to learn from the patients, to continue to learn from the patient advocacy orgs, the clinician, payers, our sponsors. And we're going to take the lessons we already have from Duchenne and our other therapeutic areas and apply where applicable, and continue to hear and understand what are the challenges and needs in DM1 that we can help meet. These are my disclosures. And so now moving into our discussion, we're going to talk about SRP-1003. And SRP-1003, it has 4 key components that are present. So this includes a conjugate or ligand, and in the case of 1003, this is an alpha V beta 6 targeting ligand. So these ligands really help guide the siRNA to that target tissue. Between the ligand and the siRNA are linker chemistries. So these chemistries really help to enhance that siRNA stability and are selected to support delivery to the intended tissue. So really helping to minimize those off-target effects. When we look at the siRNA, this siRNA has really went through a number of modifications, and these modifications are really going to help to optimize that potency, the duration, and that target specificity. So if we think about some of these all belong to what's called the TRiM platform, or the targeted RNA molecule, and this platform has undergone decades of research to really help with some of these modifications. And they include, for example, improving nuclease resistance. There's also opportunities to reduce some of that immunogenicity we see, and also to help maintain that high affinity for risk. And then we have some pharmacokinetic structures. In the case of 1003, we have a lipid moiety, and that really helps with some of those PK properties. So for instance, extending that half-life. And so what differentiates 1003 is our target. So we target our alpha V beta 6 integrin receptor. And these receptors really have some high expression in the muscle, and they also have surface availability. So at any one point, there's about 40% availability of the drug to bind to these receptors. So this makes it a very promising receptor for us to continue to further explore. We've seen from preclinical data that there are opportunities to really show robust PK properties as well as PD properties indicating a PON mechanism of action. And so to bring this all together, this is how we've talked about the distinct mechanism that we see here. So the 1003 gets into the cell through that alpha V beta 6 receptor. And once it's within the cell, it really helps to get into that RISC complex. And once within that RISC complex, binds to that DMPK mRNA, leading to that reduction in DMPK gene expression. And as we know, RISC is a multiple turnover enzyme. So therefore, incorporating that single strand of siRNA that could really help to turn over multiple molecules of DMPK mRNA and ultimately lead to that reduction of the corresponding protein. So now let's get into where we are with our study, our 101 study. So this is an ongoing phase 1/2 randomized placebo-controlled trial in patients with DM1. We have 2 parts of the study. So the first part is the single ascending dose. So you can see here where patients are first dosed with the blue dot, as well as when they undergo patient-reported outcomes or clinical outcomes. And this includes pre-dosing on day 1, day 30, 60, and 90. And in part 1, we had 2 cohorts. So we had cohort 1 that had approximately 1 mg per kg of siRNA, and cohort 2. Looking at part 2, this is the MAD portion of the study, and you could see the similar figure here where we went up to cohort 5. As mentioned, this study is currently ongoing, so we are currently enrolling in cohort 5 now. Key thing to keep in mind is we're going to show some very early work here. These are interim results, and the key focus of these early results is to show that safety and tolerability. So this is the primary of the study, is safety and tolerability. The secondary is PK and plasma. And then when we look at some of those key exploratory endpoints, we have muscle concentration, mRNA knockdown, and splicing. We also have some functional. These are some of the key exploratory endpoints, as well as some of those PROs like DM1 active C and MD high. So now going into baseline characteristics, we could see here the patients that were included in the SAD as well as the MAD portions of the study. Overall, we had 36 patients, and 11 of those patients were on placebo. Now going into what was the most important aspect of the study, and that is the safety. So we can see in the table here, this is broken up by the cohorts, so patients that were in the SAD cohort, in the MAD cohort, as well as overall. And what we saw was that the majority of those treatment-emergent adverse events were really mild or moderate in severity. There were 3 serious TAEs that occurred in 2 patients, and importantly, all were unrelated to study drug. This included a fatal arrhythmia. This included limb abscess and emotional distress. And I think when we think about that fatal, that cardiac arrhythmia, we know, of course, this is a risk when it comes to DM1. And this occurred in that first cohort and it was there was a temporal relationship where it was quite long after that initial dose. And this was, of course, heavily investigated, and it was deemed unrelated by the investigator, by the sponsor, as well as an independent coroner. The most common TAEs were headache and upper respiratory tract infections that occurred in about 13.9% of patients. And we had no TAEs that occurred over 20% of patients. So what we see here is that the TAEs did not appear to be dose dependent based on the TAE frequency at the different SAD and MAD doses, and that 1003 appeared safe and well tolerated to date in the ongoing study. Next is some early PK data. So what we see here is we see a dose-dependent increase in plasma PK, and that's up to our 3 mg per kg dose. So this is the highest dose that we have available data when it comes to plasma exposure, and it's, again, very important for us to see this dose-dependent increase, confirming that pharmacokinetic linearity. And then in terms of muscle delivery, so with the muscle delivery, this is from our low dose. So we see this is from our lowest dose, that 1 mg per kg of siRNA. And what we see is we see robust delivery into the muscle. So we know that through the alpha V beta 6 platform that our 1003 is able to enter the muscle. And we're very excited with that potential. We've seen preclinically what this can lead from a PD perspective, and of course we're awaiting to see those results as we accumulate data in this study. So in conclusion, these are interim findings. These are very early interim findings from a phase 1/2 study, and we really wanted to first show some of that safety. And what we saw was that there was no dose-dependent increases in TAE and no dose-limiting safety signals. 1003 plasma exposure increased proportionally, and it achieved high-level muscle delivery with that lowest dose, and also further showing the potential of alpha V beta 6 integrin targeting approach. These findings together, they support that continued investigation of 1003. These were really the first results. We're very much looking forward to sharing additional results as we get data from more of the patients from the SAD portion, as well as the MAD portion later this year. So with that, a big thank you first and foremost to our patients, to all the sites, and to the community for all their support with our program.
Good morning, everyone, and it's a true pleasure, and I'm quite humbled to be with you here today. Before I start, I do want to say that at Lupin, we're not only developing the treatments, but we also really try to understand the lived experiences. And one of the daily experiences that patients and people with DM1 and DM2 have, it's the presence of the myotonia. So I'm going to talk about the impact of myotonia in patients with both DM1 and DM2. So I'll share some insights from the observational studies that we have been putting in place, and also talk about our interventional study program. So this is a short disclaimer. This presentation is not promotional, and if I mention some of the treatments, some of them are not being approved in patients with DM1 or DM2. So this is purely educational and to share the research here. Lupin is a multinational global company. And this is indeed just here to emphasize that we have an opportunity with global presence to run the studies international, to involve the international experts, and ultimately the opportunity to bring the treatments faster to the patients. The Lupin Neurosciences is a dedicated franchise that does require a certain investment in the neuromuscular disorders, and again, because of the complexity of the neuromuscular conditions, this allows us to have a long-term commitment, and also to involve international experts in order to better understand lived experiences in patients with myotonic disorders, as an example. So here, I would like to emphasize that these multisystemic manifestations for both DM1 and DM2 do overlap with one of the symptoms, which is myotonia with channelopathies that is already successfully treated by namuscler, so mexilitine, that has been approved by EMA for adults. And we have recently received the positive CHMP opinion back by April that is currently under the full review with EMA for patients in less than 18 years old and starting from the 6 years old. So the innovation lies in the improved properties of the prolonged release of the mexilitine in order to investigate for impact on myotonia in patients with DM1 and DM2, so both of them. So why myotonia matters? To some of you, this work will not be really new, and this is just a representation, a snapshot of all that has been done over the decades in terms of understanding of the impact of myotonia in patients with DM1 and DM2. This is a recent study that we have put in place in collaboration with clinical experts, but also the patient advocates, a patient-tailored, patient-reported outcomes survey that we called ANSWER. It's been just recently, a couple of days ago, accepted for publication in BMC Neurology, and it does talk about not just pure impact of myotonia as one of the symptoms, but it highlights that more than 90% of patients do experience myotonia, and up to 75 at the time of filling out the survey have reported that myotonia severely impacts them. It's not just, the data do show that it's not just a single isolated symptom. It is a major symptom that is tightly connected with impact on daily activities, fatigue, but also pain and ability to perform daily tasks and work or study or being employed. So this is not an isolated symptom. It is an unmet, significant unmet medical need. And one way of thinking of the research and innovation is to look how with this prolonged release formulation, that is an experimental one, mexilitine can improve the outcomes on myotonia, and the impact on myotonia is under currently clinical investigation. So the current trial is active, and I'll give you a short update. But the idea is that the once daily formulation is clearly designed to improve not only the tolerability, but also see if this could be more effective in patients, bringing better adherence and compliance with the treatment, given that we know how complex DM1 and DM2 disease are. So again, this slide just illustrates the complexity of the multisystemic manifestations, with the emphasis that in the clinical program that we've put in place, Hercules and Atlas, that are currently running in Europe, so in EU and UK, looking at the efficacy and safety of mexilitine PR, are unique in terms of targeting both patient groups, so DM1 and DM2, and is looking into addressing one of these major unmet needs in people with DM1 and DM2. I would like to talk a little bit in detail of these two studies that we're putting in place, and we have two posters. They are already Encore posters. We've presented them at WMS meeting, so please come over and we can discuss the study design. It's the ambispective, a non-interventional study. Ambispective means that we are collaborating with the DM Scope, which is a national, most comprehensive to this point in Europe, national registry that has been collecting data since more than a decade. And it allows us to include patients into prospective study data collection that already have historical data. So it allows combining the medical history of the patients followed in DM Scope prospectively into the track DM. So we've called it track DM. And we will have several time points where the clinical aspects of the disease, including myotonia, but also all the other aspects with the patient-reported outcomes that are not only the part of the DM scope, but also are specific to the track DM to better understand the burden of the DM due to myotonia and in relation to other DM-specific symptoms and aspects of the disease. So this is not all. The core DM scope ANZA study is also there to bring the associations between what the patients say by sharing with them and French-specific, so ANZA-specific. I've just spoken about the ANZA study, which was an international one and is going to be published in BMC Neurology. So the ANZA core DM scope is about to link the clinical myotonia being measured by a clinician in the track DM with how the patients perceive the impact of the myotonia and other symptoms, including pain and stiffness, which we've uncovered also is to some degree not the same, but also fatigue and other aspects on daily activity in what physicians measure. So it does aim to improve understanding of the symptomatology of both DM1 and DM2, but also looks into the way how we can improve the awareness of the disease burden going forward, since we are looking into so many such a great treatment landscape in the upcoming 5 to 10 years in DM space. At the end, I would like to really thank the patients and families and also the study centers that continue contributing to this research, because of course without you and your input, this work would not be possible. Thank you so much.
Thank you, Lupin.
Good morning. My name is Julia Prasanas. I'm with Arthex Biotech. And Arthex is a clinical stage company headquartered in Valencia, Spain. We have a lead asset, ATX-01, which is in clinic currently in a clinical trial for DM1. Arthex has developed a boost-on platform, which is to enhance nucleic acid-based therapy delivery to muscle and brain tissues. And I'll speak about that more in the presentation. So what makes us different? ATX-01 has two differentiating factors. The first being the target. We target a microRNA miR23B, which allows for a dual therapeutic approach. And second, our delivery method. The anti-microRNA is conjugated to oleic acid, which improves muscle and brain delivery. So Arthex has taken a unique approach, and aside from MBNL that's sequestered in the toxic DMPK mRNA foci, resulting in lack of functional MBNL and resulting in aberrant splicing, there's an increased expression of microRNA miR23B. miR23B is a negative regulator of MBNL expression, therefore higher levels of miR23B, lower levels of MBNL translation. So ATX-01, by targeting miR23B, we increase the level of MBNL expression. And this resulting increase in MBNL expression leads to a correction in missplicing, including of the MBNL protein itself. And so the MBNL protein with exon 5 removed, exon 5 contains a nuclear localization signal, allows rebalancing of the MBNL isoforms between the nucleus and the cytoplasm. And what Arthex has shown, and what other groups have also shown, is that for the toxic DMPK foci to form, you need two components, the DMPK mRNA, but also the misspliced MBNL containing exon 5. So this rebalancing of the MBNL isoforms results in destabilization of the foci, allowing transport of the DMPK mRNA to the cytoplasm, where it's rapidly degraded. So what you see on this slide here is showing that reducing or targeting miR23B is a very potent therapeutic strategy. On the left-hand side graph, you have ATX-01 and GAPMA against DMPK, at a concentration that results in about 50% reduction in DMPK. And what you'll see on the right-hand side graph is at this concentration, you see a greater rescue in splicing with ATX01 compared to the gapmer. The reason being, as shown in the middle graph, that there is an increase in overall MBNL levels. So just to recap, for the toxic foci to exist, aside from the mutant DMPK mRNA, you also need the misspliced MBNL. And ATX01, through direct inhibition of miR23B, allows upregulation of the MBNL, restoring the balance of the MBNL isoforms between the nucleus and the cytoplasm. And this destabilizes the mutant DMPK foci through recovery of this correct MBNL isoform pattern, leading to foci decay. So for further details on the mechanism of action and all the work that went into elucidating that, please see poster 129 and discuss with my Arthex colleagues, who'll be happy to take detailed questions on this and the work going into it. So we've shown previously that ATX01 rescues functional and molecular phenotypes in a mouse model of DM1, the HSALR mouse model. So after multiple dosing, we see regain of normal strength, which is sustained, dose-dependent splicing rescue, and preferential MBNL elevation in muscle, heart, diaphragm, and brain. These results can be seen in the Gonzalez-Martinez paper from this year. In a second mouse model, the DMSXL mouse model, inhibition of miR23B corrects neurological defects seen in this model. So again, after multiple dosing, we see MBNL levels increase in the brain temporal region. We can detect the compound in the brain, and the bottom right-hand graph shows a rescue of the anxiety-like behavior that can be seen in these mice. So with an in situ hybridization, we can see that ATX01, the green signal, co-localizes with neurons in yellow signal and astrocytes in red on the right-hand side. It reaches all areas of the brain, including the cortex, cerebellum, and the spinal cord on the right-hand side, but it also reaches subcortical levels of the brain, as you can see on the bottom left-hand side. So our first in-human study is underway in Canada, the US, UK, France, Italy, Spain, and the Netherlands. Many of our sites are in the audience. As a first-in-human study, the primary objectives are safety and tolerability. We are looking at target engagement in the muscle via various biomarkers, including MBNL levels and splice index. And we have a number of exploratory objectives looking at clinical endpoints, including muscular, extramuscular function, and patient-reported outcomes. So it's a randomized, double-blind, placebo-controlled, single and multiple ascending dose study. So our part one of the study is the single ascending dose. We have 4 dose levels. In each dose level, we have 8 participants randomized in a 3-to-1 active to placebo ratio. After a single dose of ATX01 or placebo, they're followed up for about 3 months. We have completed dosing in the first 3 cohorts, and we are currently dosing our 4th cohort. Dosing in the 3rd cohort in the single ascending dose allowed us also recently to start dosing in the multiple ascending dose portion of the study. We have 3 dose levels in this part of the study, starting at dose level 2, which is currently dosing now. So our inclusion and exclusion criteria briefly, we have patients with more than 150 CTG repeat lengths and myotonia of above 3 seconds. Key exclusion criteria include congenital DM1 and taking of anti-myotonia medication. Our key outcome measures, safety and tolerability, obviously, and then we have a number of muscle and function testing and clinician and patient reported outcomes to try and get as much data as we can from this phase 1/2 study to help inform future studies. So we have some key milestones recently achieved, including starting our multiple ascending dose. We hope to have readout from the part 1 of the study at the end of this year and readout from part 2 of the study at middle of next year.
All right, good morning. That was a long walk up here, almost the same as going up 175 from Quebec City. And I actually bumped into my colleague there, sorry about that. So I'm Jeff Haroldson, representing Avidity Biosciences. Avidity is a San Diego-based company. I think maybe the biggest news for us this year is in February, the acquisition deal closed from Novartis. So now we have a significantly bigger footprint globally, and I think that infrastructure and execution is going to be very, very promising to have much more dramatic global reach for patients. Forward-looking statements. I'm not going to read all those. The one thing that, I was thinking about this that really hasn't changed. I mean, we're actively integrating now, but the one thing that really hasn't changed is the vision, and really behind leveraging our RNA technologies for these patients like Luke, who, not unlike other individuals with DM1, went through a dramatic diagnostic odyssey, taking almost 8 years to get an accurate diagnosis. So there's still such a dramatic unmet need in this population. So our product is an AOC, an antibody oligonucleotide conjugate, really leveraging 2 well-known and established technologies, monoclonal antibodies and oligonucleotides, and putting them together where the sum is greater than the whole of its parts. So in this case, oligonucleotides target RNA very well, but they've always struggled getting beyond the liver. So in this case, that monoclonal antibody targeting transferrin receptor 1, which is on muscle tissue, skeletal muscle, cardiac muscle, and smooth muscle, like in the GI. So we see a lot of potential great benefit there. The whole complex is, once it hits the receptor, it's taken inside the cell, and then that double-stranded siRNA loads onto the RISC, or the RNA-induced silencing complex, and that passenger strand then is cleaved, and the guide strand follows its complementary sequence on the DMPK messenger RNA, where it then degrades through hybridization of the toxic messenger RNA. We have a very robust clinical development program. I think the one thing here that really is amazing to me is that the phase 1, phase 2, our MARINA study started, as you can see, in 2021, and we are now in 2026, and we're ready to complete our global phase 3 Harbor study towards the end of this year. We're shooting for top-line data readout in the second half of this year in 2026. So very aggressive movement, and we have done that with our other programs as well, not just DM1, with DMD and FSHD equally. The phase 1, phase 2 MARINA, of course, is primarily a safety and tolerability study. Patients did really well in that trial. We actually now have some patients that have been on drug for over 4 years, and we had 1 SAE withdrawal. It's been, I think, publicized very well and discussed, and in fact, in a recent publication of the MARINA trial design and key results in the New England Journal of Medicine this year, that's discussed in there in good detail. So we basically looked at a single and multiple ascending doses from 1 to 2 to 4 milligrams per kilo, and ultimately we landed on a 4 milligram per kilogram every 8-week dosing. So the MARINA patients, all those eligible 37 patients, moved into the OLE, or the open label extension, which is now all those patients have moved into our Harbor open label extension. So that's the evolution of our clinical development program. We have had a good year so far. We've had some really key publications. As I mentioned, the MARINA study, the final study data is published in the New England Journal of Medicine. And then we had another publication in Molecular Therapy with our scientists at Avidity, demonstrating the molecular pathology of disease with DMPK knockdown, MBNL, increases, the sorts of conversations we've been hearing earlier. And with those early increases in MBL, even seeing later correlations with some functional outcomes that we are assessing in trials such as VHOT and QMT. So Marina, our phase 1, phase 2, really well tolerated. Secondary proof of concept, DMPK knockdown, an increase in functional MBNL, and then the exploratory endpoints as I had on another very busy slide. Hand myotonia, hand grip strength, QMT, and our PRO that we're assessing, DM1 active. So where do we go from here? We are in the Harbor Global Phase 3 program now. That enrollment was completed in July, and we're targeting top-line data at the end of this year, in the second half of this year, which is very exciting. And we carried through those same assessments. It's not listed here in that 4 milligram per kilogram treatment arm versus placebo. The biggest change that we did make as a result of the acquisition as well is we originally had a 30-week primary readout. We've moved that to 54 weeks. And so we've sort of pushed everything about 6 months and allowing for demonstrations of greater durability and looking at those same assessments. It's a big thank you to everyone who's been involved. I know a lot of you out there are involved and have been involved in Marina and through to Harbor. All the investigators, study staff, patients. Obviously, we can't do any of this without everyone participating. So we're excited to see what the end of this year looks like for us with a whole new top-line data release.
Thank you very much.
Hello, everyone. My name is Allison Cruiser, and I'm very honored to be here to speak about Dyne Therapeutics and our DM1 program. Thank you so much to MDF, to Euro-Dyma, and to the IDMC organizers for having us here today. And even though I'm here to speak about DM1 and very honored to do so, I do want to make a quick announcement on behalf of Dyne. You may have seen, if you were following our news, that we did have a milestone moment for the company today with the submission of our first BLA to the US FDA. So we are very excited about this. It is actually a BLA. Thank you very much. A BLA for our investigational drug called Z-rostudirsen, which we hope will be a drug that will be approved for the indication of exon 51 skipping and Duchenne muscular dystrophy. So a very important day for Dyne, and I'm thrilled to continue it to speak about our DM1 program, focusing on our late-stage clinical development in phase 3. These are our forward-looking statements. At Dyne, we are on a mission to deliver functional improvement for individuals, families, and for communities. And truly, what does this mean? So we believe that it is very important to be listening to the patient community, to patient advocates, and to leading physicians understanding what the true impact of DM1 is. And we have developed a platform called our FORCE platform that we believe is uniquely equipped to hopefully help address some of these many multi-system manifestations of DM1. So what is the FORCE platform? So this is predicated on a proprietary fragment of an antibody or a Fab, similar to my colleague Jeff from Avidity, who spoke about leveraging the internal power of the TFR1 receptor. Our FORCE platform also is enabled to do this. And what we hope is that by utilizing the fragment of the antibody, which is represented by, in the cartoon, about one side of the Y of the antibody, if you look at the zoom in, is that we hope that that small version of that TFR1 recognizing antibody is able to more diffusely penetrate throughout the different tissues that matter throughout the body. And in addition, we hope that the size will allow for it to have very supportive internalization kinetics that will not interfere overall with the endogenous role of the TFR1 receptor inside the body. And we'll talk a bit more about our early clinical data and what we've learned so far with a favorable safety and efficacy profile to date. In addition, we have a linker, and then importantly, we have a modular payload, and the modularity allows us to be able to utilize the FORCE platform to be able to hopefully address different rare neuromuscular diseases that are genetically based. And in DM1 in particular, why we are so excited about the potential power of the FORCE platform is its ability to target both skeletal muscle, including the diaphragm, smooth muscle, cardiac muscle, and the CNS. So I won't talk as much about the CNS today, but our Chief Science Officer, Ron Batra, who many of you know, will be here later tonight, and he would love to speak in much more depth about our preclinical CNS data. But I do want to say briefly that we believe that our fragment antibody that targets TFR1 can mediate transcytosis across the blood-brain barrier, as you can see in the cartoon on the left-hand side of the screen. And just one brief piece of preclinical data from non-human primates shows that this is indeed the case, and that we can see on the right-hand side of the screen very diffuse ASO staining if you look throughout the brain if we utilize the FORCE platform to deliver a gapmer ASO versus naked ASO with no FORCE platform has very little or no staining. I certainly don't need to talk about the history of DM1 speaking in this room. I will say that, as you all know, it was about 1900 when Hans Steiner first defined or clinically defined myotonic dystrophy. And it took about 100 years for many of the people, again, in this room and many of their proteges to really define the molecular pathophysiology, which is just one of the reasons why it's an honor to be here speaking in front of you today. And what has also very much supported the field of clinical development was work done by the Thornton Lab and others in 2012 that showed that there was a symptom, myotonia, that could rapidly be responsive to targeted genetic treatment. And this really helped us understand as a field and as the biotechnology companies how we might be able to generate clinical trials that could show a functional response in a shorter amount of time and hopefully be able to bring therapeutics to people as quickly as possible. And so you see the clinical development really launched in 2014 for DM1, and in 2021 and 2022, Dyne was founded, and as well as other companies here, have started thinking about how to use conjugated therapies to really be able to solve some of the delivery problems that my colleague from Avidity also described very, very beautifully. And so here we are today with some late-stage clinical trials, but not nearly as much as we'd like to see in the field of DM1. We are really proud to be starting our phase 3, and we hope that this will be a phase-defining phase 3, and we'll talk a bit more about it. But we do want to note that we recognize that all of the work that we are attempting to do through our studies has really been standing on the shoulders of you all. So we do have a poster later this week, which will go through much more depth of the early proof of concept safety and efficacy data from our drug, zeliciment basavarsen, or Z-basavarsen, which you may previously have known as Dyne 101. So I won't go into too much data today, but I do hope that you have a chance to check out our poster and to speak to our clinical development lead, Dr. Shanna Anderson, who will be presenting that. But briefly, our multiple ascending dose study for ACHIEVE was our first in human phase 1/2 clinical trial. We dose escalated and we selected the final dose of 6.8 mg per kg, which we're moving forward with in both our ACHIEVE registration expansion cohort and also our phase 3 study, which I'll talk more about in a moment. So participants from all dose cohorts were eligible to enter an open label extension and in a long-term extension, which is still continuing today and continues to lead very informative data for us on safety and efficacy of long-term treatment. This is just one high-level overview of the data. Again, I highly recommend that you speak to us at the poster session where we can go in much more depth. But we're very pleased to see trends toward functional improvement across a variety of clinical measures. Again, as you know, DM1 is a complex multi-system disease, and so we are trying to work through our endpoint strategy to make sure that we can adequately represent, to the extent possible, all manifestations of the disease while still maintaining homogeneity so that we can see clear distinctions so the treatment be working as expected. So if you look on the screen, when you see where 0 is, that's baseline, and if someone stayed at 0, that means they stayed exactly where they started. You can see that in the gray, which is representing 6 placebo patients who were treated with placebo for 6 months, largely across the functional measures, they tend to get a little bit worse or stay close to where they started. The blue bars represent people who were treated with the 6.8 mg per kg go-forward dose of Z-basavarsen. There were 6 of those participants who were treated with 12 months, and you can see that largely we are seeing trends in the positive direction. And this is across a number of different functional measures, so VHOT, which we know is a measure of hand myotonia and function, also time function tests such as the 5 times sit to stand, 10-meter walk-run, as well as strength, fine motor through the 9-hole peg test, and also importantly, what patients say is being impacted by them. So we leverage several PROs in our clinical studies, but we find it very important to utilize the MD-High as well, which was co-developed with patients in the University of Rochester and many investigators who are here. And it covers 17 different domains of patient functioning, again, through co-creation with patients as they were telling drug developers really what they should be looking at to understand whether or not the impact of disease was improving for them. So we're very pleased to see that we're seeing positive trends in MD-High through this treatment as well. And of course, as we also know, the core molecular pathophysiology for DM1 is splicing. So to understand whether or not our trends in efficacy are working because our drug is working as intended, we are very pleased to see that splicing is also corrected over time. So this is one brief overview of what we were seeing for our splicing pharmacodynamic profile from ACHIEVE. So this is the CASI-22 mean. This is a 22 gene panel that is known to be, 22 genes that are known to be impacted for splicing for DM1. So where you see the blue bars, again, those are the participants who were treated with 6.8 mg per kg over time. These are biopsies from the TA, and you see that over time you see consistent trends towards improvement in splicing, versus those treated with placebo over 6 months see slight worsening of splicing or near baseline of splicing. So overall, we were very pleased with that early proof of concept safety and efficacy data from ACHIEVE. And as I mentioned, we are continuing to follow patients in the multiple ascending dose long-term extension to get additional long-term data, which we look forward to sharing with you in the future. But these data also made us realize that we need additional patients and we need a larger powered study to better understand whether or not this drug is working as intended. And so that is indeed what we are running with the ACHIEVE registrational expansion cohort. This study is ongoing to date. There are many investigators in the room, and I want to say a huge thank you to all of the investigators and site staff who are working so hard on the MAD portion of ACHIEVE, but also in the REC. We announced recently that this REC portion has enrolled its target of 60 participants. We are continuing to work through the screening funnel of participants, and we hope to close enrollment completely by Q2 of this year. And should we remain on track, which we expect to do so, we would have top-line readout of our ACHIEVE registrational expansion cohort in Q1 of 2027. And should that be favorable, we hope to work with the FDA to potentially file an accelerated approval application. But we don't want to stop there. It's not just about filing for accelerated approval in the US or about the ACHIEVE study. In addition, we want to make sure that we are really truly looking at multiple multi-system manifestations of DM1. And so we have already initiated our phase 3 study, which is called Harmonia. So the goal of Harmonia is to assess and confirm the multi-system functional improvement and also to support global access and reimbursement should our data be favorable. And we do want to say that we are very pleased because we know that we can't design clinical trials in isolation if we want them to actually generate data that meaningfully report back on whether or not the drug is improving patients' lives. And so we're very thankful for the advocacy community, patients, and many leading physicians who have supported us in trial design. So VHOT is a very important measure to give us early indication of whether or not our drug is correcting underlying splicing. And Jeff, I think you did a beautiful job explaining that earlier in your talk. It is reliable and responsive. We don't believe that it is the most functionally meaningful endpoint for individuals, and we have heard from many patients that for many patients it is not as applicable to activities of daily living. So as we started to think about late-stage clinical development for zeavazovarsen, we started to think about what might be an appropriate endpoint that would help inform additional information, additional data that we're generating from the ACHIEVE study, and also kind of hit those last two markers of functional meaningfulness and applicability to ADLs. And we've landed on an endpoint called the 5-time sit-to-stand test. So if you look in the middle, this endpoint is fairly simple from the graphic. People sit in a chair without using either their arms or arms of a chair, and they sit and stand 5 times. And the ability to complete this and the amount of time that it takes to complete this is measured through the 5-time sit-to-stand process. This provides a unique amount of information from one endpoint. So we are able to get information on lower extremity function, balance, functional mobility, stamina, trunk muscle engagement, and very importantly to us, we also know that it's relevant to activities of daily living. We know that people are sitting and standing and transferring throughout many aspects of our lives. And so we believe that this might be a measure that can help report on independence and autonomy, which we know is absolutely critical in the DM1 community. And if you look on the left-hand side of the screen, 5-time sit-to-stand and other neuromuscular diseases has been shown to assess fall risk. We know that falls are critically important in DM1, and the field probably couldn't speak about the impact of falls enough. We know people have an increased rate of falls, but even in speaking with a person impacted by DM1 just recently, she mentioned that even if she's not falling, she's changing her behavior because she's afraid of falling. And I thought that was really powerful to think about the overall impact, not just the falls and not just the injuries, which of course are critical to people who are living with DM1, but also the ability to go out and to live your life the way that you would like to live that without the fear of falling. And then on the right-hand side of the screen, you can see that 5-time sit-to-stand also requires engagement of trunk muscles. And to this crowd, I don't need to say much more about the importance of trunk muscle strength, but we know that trunk muscle strength helps support not only balance and postural control, but also helps support respiratory muscles, which are critical. So taken together, these are some of the rationale around why we have selected this test as a primary endpoint for our phase 3 trial. We're also very thankful to Dr. Charles Thornton and other investigators who have looked at the 5-time sit-to-stand and DM1 natural history, and analyzing the data set that they generated has been really critical for us as we've thought about designing our phase 3 clinical study. So just very briefly, the DM1 impact on 5-time sit-to-stand is profound. So we know that by looking at natural history data, there is impairment for people living with DM1 detected at each decade that we analyzed compared to individuals who are not living with DM1. And not only that, but people who are living with DM1 have impairment that can be 5 to 9 seconds slower than people living without DM1. Again, thinking about age-matched controls over time. And we know that there's an accelerated rate of decline across the decades of life. So together, we believe that this shows, it helps lend evidence to show that this could be an appropriate endpoint for DM1, and then it might be something that really could help meet those additional barriers that I mentioned earlier that we'd like to check have functional meaningfulness for patients and applicability to activities of daily living. And so our hope is that this endpoint will provide a comprehensive functional measures of these outcomes in DM1 clinical trials. So just briefly with the study design, I will be presenting a poster later this week where we go into a bit more detail. So again, would really welcome you to come up and speak during the poster session if you want to talk about the details of Harmonia. But very briefly, we'll be enrolling approximately 150 participants. Participants will be ages 16 and up with no upper age limit, assuming that people meet the other eligibility criteria. They'll be 1-to-1 randomized for 48 weeks to either Z-bazavarsen 6.8 mg per kg or to placebo delivered by infusion of Q8 weekly, followed by a long-term extension. And in addition to the primary endpoint of 5-time sit-to-stand, which I just spoke about for several minutes, we will also be looking at a variety of secondary and exploratory endpoints, which I'll show in just one more slide. So briefly for the key inclusion and exclusion criteria, I mentioned the age of 16 years and greater. Also, we are using a repeat size of greater than 100, and individuals need to be able to complete the 5-time sit-to-stand and the 10-meter walk independently. We are excluding individuals who have a known diagnosis of congenital DM1. Importantly, we are able to include individuals who have diabetes or who have a pre-existing pacemaker or defibrillator. That was something that was an exclusion from our phase 1 trial. And so as we're thinking about how to maintain homogeneity in our phase 3, but also try to expand criteria where we can, we are pleased that we were able to do this, working with physicians and patients to think about how to do that appropriately. Okay, so what are some of those additional endpoints? So as I mentioned, it's very important to Dyne to be thinking about the multi-system manifestations of disease. So we have selected a variety of endpoints. And some of them you've noticed from the previous slide where I talked about achieve from the multiple ascending dose portion. So VHOT, QMT total, 10-meter walk run, MD high, additional PROs, all will be included in our secondary endpoints. And also importantly, I started the talk by saying that we know that there is a profound impact of the CNS upon the lives of people who are living with DM1 and upon their families. And this covers a range of different domains: cognition, behavior, emotion, fatigue, sleep, emotional daytime sleepiness, etc. And this can be a very heterogeneous and very impactful set of symptoms for people living with disease. And we are working very closely with the physician and patient community to understand the best way to measure the impact of our drug upon some of these different domains of health. So to begin, for our phase 3 study, we are including a range of endpoints and exploratory measures. And I thought this was interesting in a way because we don't always talk about exploratory measures in biotech and biopharma. And usually that's because for exploratory measures, it might not be included in some of the powering or the way that you explain the study. It might not be considered in some of your inclusion-exclusion criteria. But I was really proud that at Dyne we all agreed that we wanted to share these and speak about these because we want to understand the impact of our drug. And we also want to learn more about these endpoints so that we can help contribute to all the work that you all are doing as well to try to understand what CNS endpoints may be appropriate in DM1. So we are measuring a range of these endpoints, including the MD high subscales, which is also an achieve, which does give patient-reported impact of disease burden on CNS-related symptoms. in addition to biomarkers, actigraphy, which could provide information into sleep, which we know is critical, also circadian rhythm, activity, perhaps a read-through to a number of different symptoms that one could consider. We'll also be measuring cognition and fatigue and excessive daytime sleepiness. And in summary, we want to say thank you again because Harmonia has been designed alongside the DM1 community. We are pleased to be using a 5-time citistand as our primary endpoint that we hope will continue to provide even more information than some of the primary endpoints that we're using in our earlier stage clinical trials. And we, in addition, hope that the broad set of secondary and exploratory CNS endpoints, as well as other exploratory endpoints, will help us really truly be able to evaluate the impact of Z-vasovarsin. We do have a number of sites that are being initiated now for Harmonia. We do have sites that are currently enrolling in the United States and Japan, and we are very actively all over the world, even this week, hoping to initiate many additional sites to continue to expand our geography. And again, I just want to thank the organizers for having us here today, and also importantly to thank every person here who has been involved in Achieve and Harmonia and provided input to our studies, which is so critical for us. Thank you.
Bonjour à tout le monde. Je suis bien content d'être ici. I'm very happy to tell you about the results from our studies that are ongoing today. I'm an employee of PepGen. These are my disclosures, and these are my forward-looking statements. So when we started PepGen, we were very much focused on the treatment of neuromuscular diseases, and in particular, have always had a focus on myotonic dystrophy type 1. And it's through our engagement with the community, bringing patients into the company, hearing their stories and understanding what they're experiencing, that we hope to do a better job in delivering a therapy that hopefully makes for a better future, along with all my colleagues gathered in the room today. So I won't spend a lot of time on this, and I'll skip through some slides just given we'd like to allow some time for questions. But just focusing on the fundamental technology, it's highlighted here schematically in the top left. We're using a linear peptide that has been specifically engineered to allow better uptake into cells of therapeutic oligonucleotides. And it consists of a variety of different component parts which both allow better cell association and, very importantly, better endosomal escape, which is a fundamental issue for all oligonucleotides, which may be able to get into the cell, but if they don't have a mechanism of getting out of the endosome, essentially will not be functionally relevant. We're showing here on the right-hand side a DM1 patient cell in vitro, and the red indicates the oligonucleotide, and we can see very, very good levels of delivery, not just into the cell, but importantly, also into the nucleus itself. And in fact, we can go and measure our ability to go and deliver oligonucleotide into the nucleus. This is an in vitro assay with DM1 patient cells, and you can see compared to naked oligo, which are the dots along the baseline here, we see a dramatic uptick in terms of nuclear delivery. So this is not simply the ability to get into the cell, but very importantly to get into the nucleus. And that's the site of action where we need to be able to deliver our therapy, because that's where the toxic foci are located, and that's where we must liberate MBNL1 protein to restore normal splicing in patients. Now, in addition to our unique delivery technology compared to what other approaches that you've heard about so far, another component that is unique is that we are not seeking to degrade indiscriminately DMPK transcript. So 2 different transcripts are being made, as was intimated earlier. The normal DMPK transcript present both in the nucleus as well as in the cytoplasm, and then the pathogenic species that forms these stem loop structures, which act as a molecular sponge for MBNL1 protein. These pathogenic structures are located in the nucleus, and before I get the question, we don't use RISC. And so our approach is instead to go and bind specifically to the CUG repeat expansions, dislocate MBNL1, and destabilize the stem loop structures. And we've been able to demonstrate across both cell-based systems from a variety of different patient cells the ability to reduce the number of foci and return normal splicing, and in mouse models been able to demonstrate restoration of normal splicing and correction of the myotonia phenotype. On the basis of this and a large amount of preclinical data we advanced into a phase 1 study. We reported these results out last year, so I'll be very brief in my statements on these. This was the FREEDOM clinical study. This was a double-blind, placebo-controlled dose escalation of 5, 10, and 15 mg per kg. Each group had 8 individuals randomized 3-to-1 active to placebo. This study is now completed, and patients from the study are now rolling over into the open label extension. One of the things that we're very pleased about with this study is that we saw very high levels of splicing correction. In fact, we saw the highest level of splicing correction, even after a single dose, than's ever been reported with any approach, even following multiple doses. We saw 12% with 5 mg per kg, 29% at 10 mg per kg, and almost 54% correction of splicing using the 22-gene panel described by Charles Thornton and others. And importantly, of these participants, 87.5% of participants showed an improvement in splicing, which we're very pleased about. And so on the basis of this and a very good, well-tolerated safety profile, we advanced into the FREEDOM-2 multiple ascending dose clinical study. This study is currently open in Canada, UK, New Zealand, Australia, and South Korea. We recently reported that we've now completed enrollment of the second cohort at 10 mg per kg. And I'll tell you in a few moments about the results from the first cohort, 5 mg per kg. Keep in mind, this is the starting dose level for this study. We hope to be advancing into the third cohort, 12.5 mg per kg, later this summer or early fall, and reporting out the results from the 10 mg per kg cohort later this year. So this is the demographics in terms of the 5 mg per kg cohort. We remain blinded to certain aspects of the study, including which of these patients are active versus placebo. In terms of the safety profile, the drug was generally very safe and well tolerated. All treatment-emergent adverse events were mild or moderate in nature. All of these were transient in nature. None required any intervention. Importantly, for this class of drugs, we did not see any noteworthy kidney treatment-emergent adverse events. There was no hypomagnesemia observed in these patients. We were, I will say, disappointed when we initially saw the splicing results, where in the placebo group, we saw a 6.8% improvement in terms of splicing in the 2 patients. Quite a bit of variability, as you can see in the individual patient data in this central panel here. And then in the active group, we only saw 7% splicing improvement. And to remind you, we saw 12% improvement in the single ascending dose study. Now, one thing that's quite noteworthy is that of the 6 active patients, we had 1 individual, highlighted by the gold bar here, who showed a 70% worsening of splicing, which is something we'd not seen previously in our single ascending dose study. The rest of the group, excluding that 1 outlier, showed pretty solid levels of splicing improvement, 22.9%. But this splicing outlier obviously skewed these results significantly. Now, interestingly, when we went back and looked at the natural history data, and this is data that's been published from Charles Thornton and colleagues in a Lancet Neurology paper, what's quite interesting is if you look at this change in splicing in patients, about 74% of patients show a movement of splicing over a 2 to 3 month period of time of somewhere in the neighborhood of minus to plus 20%. That's where most patients show the natural movement in terms of splicing. Relatively few patients show an improvement of greater than 20%. In fact, only about 1 in 10 patients show better than 20% improvement. Now, interestingly, in this natural history data, you can go and see a few patients, I think it was 2 or 3 patients, who showed a 70% worsening, and presumably that's what we saw in our MAD data set. But it's noteworthy with this data that I think if we want to really fundamentally move the needle for patients and be able to show a meaningful change, we've got to do better than the 74% of patients who are basically showing movement of splicing of plus or minus 20%. And so as we go and look at the right side of this curve in the context of the data we've presented from both the single ascending dose study as well as data from the MAD study, we can see that the placebos and the MADs for both of these studies are in that plus or minus 20% range. But in the single ascending dose study, we can see at 10 mg per kg and 15 mg per kg that we actually see an improvement beyond that, whereas both of these groups showed meaningful correction greater than 20%. And it's interesting, not only did the majority of patients show improvement, we can also say that the majority of patients in these two groups showed a greater than 20% improvement in splicing. We've looked at functional outcome measures, and again, this is our starting dose level at 5 mg per kg, and we did see an improvement in VHOT, but as others have noted, VHOT is a highly variable outcome measure, and we had one patient in particular. Interestingly, it was the same individual who showed the splicing variability of 70%. This individual showed a 22-second difference in his VHOT results from his nadir at week 8 to week 16 and drove much of the return to baseline we saw at week 16. But for most time points, we saw an improvement. And indeed, as we dive into the individual patient data, we can actually see the majority of patients saw an improvement compared to baseline at all starting points. We hope to be able to build upon this with our 10 mg per kg and our 12.5 mg per kg dose cohort. And based on the splicing results from the SAD, we think that's a reasonable expectation. And so just to wrap things up and allow time for questions, what's got us very excited about what we might be able to do in terms of improving this disease for patients is that we've developed a truly differentiated delivery technology that allows very good nuclear delivery of oligonucleotides. We're targeting specifically the CUG repeat expansion RNA that drives this disease, as opposed to focusing on the non-pathogenic RNA that exists in the cytoplasm. And to date, we've demonstrated a very good safety profile for this and look forward to provide a series of updates later this year, including we'll be providing a safety update once the DSMB has met to review the unblinded data from the 10 mg per kg cohort, which has now been fully enrolled, and then providing further updates on the 10 mg per kg, including functional outcome measures later this year.
Hi, good morning, everybody. I'm Alison McVie-Wiley from Vertex, and before I begin, I'm actually going to just spend a couple of minutes after my disclaimer to introduce Vertex. So who are we? So Vertex is a global biotechnology company that has the goal of creating transformative medicines for serious diseases using cutting-edge innovation. As you've heard from, I think, all of the companies this morning, patients are really the heart or the center of everything that we do. So every decision that we make is influenced by patient voice, and we operate with a sense of urgency because we know that every day matters. At Vertex, we strike at the core of serious diseases, and what we mean by that is that we really invest time to understand the human biology of the diseases that we work on, and then we develop therapeutic approaches to really target the root cause of the disease, not just the symptoms. And we're not afraid to take on challenges. We work on a number of challenging and tough diseases, and we don't rely on what we've done in the past. We really focus to develop the right approach or the right tools to treat the particular disease that we're working on. You can see that, I think, from our diverse pipeline, in which we work on multiple serious diseases, and you can see that we really work on a number of different modalities, from small molecules to ex vivo and in vivo gene editing, to oligonucleotides, as well as cell therapies. As you can see for myotonic dystrophy type 1, we're actually evaluating 2 approaches to treat DM1. One is our oligonucleotide therapy, and I'm going to talk today about our first asset in our oligonucleotide program. But we are also researching potential small molecules for DM1 that would also potentially target the root cause of the disease. So moving on, I'm going to introduce our investigational peptide conjugated oligo for DM1. I don't think I need to go through this slide with everybody in the room, but really suffice to say, because the molecular basis of DM1 is very well understood and very well characterized, then it really fits very nicely with Vertex's R&D strategy, and that it allows us to design potential therapies that really target the root cause of the disease. And our approach to that is actually to address the underlying cause by targeting the toxic RNA hairpin with a steric blocking oligonucleotide. So in some respects, actually a very similar oligonucleotide mechanism that you just heard from PepGen. I think one of the differences in our approach is in our delivery system. And this is our lead asset for DM1 in the oligonucleotide space. This was an asset that actually Vertex licensed at the end of 2022 from Entrada Therapeutics. You can see that one component of our molecule is the PMO that functions as a steric blocker and blocks the muscle-blind sequestration to the toxic or the pathogenic RNA hairpin. And that's linked actually to 2 peptides. One is a cyclic peptide and one is a linear peptide. The cyclic peptide is designed to get the oligonucleotide into tissues and out of the endosome. The linear peptide is designed for nuclear translocation to get the PMO efficiently into the nucleus. This just highlights, in terms of our mechanism of delivery, as mentioned, the cyclic peptides really induce high efficiency intracellular delivery into tissues throughout the body, and they do that through the endocytosis pathway. So once that molecule is in the endosomes, there has to be a mechanism by which the oligonucleotide can then get out of the endosome. And it does that through the design of the cyclic peptide, which induces budding from the endosome and can release a high proportion of oligonucleotides then into the cytoplasm, and then that nuclear localization signal can then take the oligonucleotide into the nucleus. So you can see that our molecule is designed to address all of the barriers that these molecules face when they're administered intravenously into an animal or into patients to get from the bloodstream to the nucleus where the pathogenic RNA is located. Once inside the nucleus, as mentioned, the oligonucleotide then selectively engages the pathogenic RNA allele with the goal of releasing muscle-blind to correct the splicing dysregulation. How do we know that that works? We have a suite of preclinical data that's shown in this slide that tells us that our molecule can reduce the RNA foci in patient cells in vitro. And as a result of that, we can also see the correction of missplicing in a dose-dependent effect, again, in patient cells. And we've looked actually at multiple patient cell lines with different numbers of repeats included. We then looked at one of the DM1 mouse models to show that following an intravenous or systemic administration, that we can also correct missplicing in the mouse in a dose-dependent manner. And one of the functional consequences of DM1 that we can evaluate in this particular mouse model is myotonia. And you can see that nice corresponding reduction or resolution of myotonia in our mouse model following treatment. So really it was this data, in addition to our comprehensive toxicology or safety package, that really supported us moving our molecule into the clinic. So where are we in development? We have our ongoing study, it's called Galileo. It's a first-in-human, placebo-controlled, single and multiple ascending dose study. For a first-in-human study, the primary endpoint is, of course, safety and tolerability, as well as looking at pharmacokinetics and pharmacodynamics in the study that could tell us whether this molecule is distributing the way that we think that it should, and also has a potential effect on splicing through muscle biopsy collection, as well as we also have included in this study a suite of functional assessments to look very preliminarily at efficacy. Part A of the study is a single ascending dose part of the study. That part of the study is now complete. We are now in the multiple ascending dose portion of the study, and we are on track to report results from this study and the completion of this study in the second half of this year. This study is a global study, and you can see that sites are active and recruiting patients in North America, the UK, Europe, and Australia. So before I wrap up, I actually wanted to spend a little bit time on our values at Vertex. So this is this is complementing what we do with how we do it. You can see at Vertex we have 4 core values. The We Wins really speaks to our outstanding diverse teams that are really tasked with solving tough problems, and doing that through innovation, and doing that using cutting-edge scientific innovation to develop the right tools for the diseases that we work on. And we always chase excellence in everything that we do. And as mentioned, patients are at the center of what we do, and that's through our value of uncompromising commitment to patients. So how do we build that value into what we're doing in our DM1 program? And that is really to incorporate the voice of the DM1 community in our ongoing oligonucleotide program, but also to help inform us of subsequent programs that we are pursuing and that we will develop in the DM1 space. We do that by investing to understand the burden of the disease, by listening to patients and patient feedback into our clinical trial design, as well as our patient-facing materials. And we do that through listening to, for example, the DM1 Community Advisory Board. And as Elaine mentioned earlier, we actually recently met with the Euro-Dyma Pharma Dialogue Group, which was extremely useful. And of course, working to educate and raise awareness of this disease. With that, we just wanted to provide some examples of some of the work that Vertex is doing with the community in terms of really helping us understand the real-world impact of DM1. And you can see that we have multiple real-world research partnerships, and actually these are with many of you that are in the room today. So we participate, like many companies, in NDM1. We also are working with DM Scope and the French National Registry to really help us understand the mortality impact in DM1, as well as the healthcare burden. We have a really amazing partnership with the team here in Saguenay, and we're so happy to be here in person for this meeting, working in terms of natural history study, and with the RNA Institute from Albany on the natural history of splicing. Some of that data is actually going to be presented here this week at IDMC in oral presentations and also in posters. Similarly, you'll hear a little bit later on this week about our partnership with Muscular Dystrophy Canada, which is really to help us understand the patient journey of people living with DM1 in Canada, as well as the impact and the importance of genotyping for an early diagnosis or a rapid diagnosis of the disease. We also work with the Swedish registry, again, to collect long-term or to evaluate long-term data and help us understand the progression of the disease through decades. And finally, we have a really nice partnership with a group in Leuven in Belgium, where we're looking at MRI and the utility of using muscle MRI to help us study DM1. So with that, I'm just going to end today with some key takeaways that at the end of the day, Vertex is a company that really invests in scientific innovation to develop therapeutic medicines for people living with serious diseases. And we are incredibly grateful for the work that we are able to do and partner with the DM1 patient community and other advocates, including Euro-Dyma here today and the Muscular Dystrophy Foundation.